Welcome to the MGH brain-PBM clinic round. Today is January 31st, 2025. I'm Paolo Cassano. It is my great pleasure to welcome and invite Dr. Nghi Truong from the Postdoctoral Researcher at the Advanced Neuroscience Imaging Research Lab, Department of Radiology at UT Southwestern Medical Center. She's going to talk about EEG microstates and photobiomodulation. Thank you, Dr. Truong, and please take it away.
Thank you for the kind introduction. Let me share my screen and then we can get started. Can you all see my screen?
We can.
Okay, thank you. Thank you. And then good morning. Thank you for having me today. My name is Nghi Truong and I'm a postdoctoral researcher at UT Southwestern Medical Center. Today I will be presenting our study on the neural effect of transcranial photobiomodulation on EEG microstates. So this work was conducted at the Biophotonic and Neuroimaging Lab in the Department of Bioengineering UT Arlington under the supervision of Dr. Liu. So I have nothing to disclose for the on the conflict. Sorry, I have no financial disclosure or conflict of interest with the presented material in this presentation. So first of all, let's start with a brief introduction of tPBM. So tPBM is a non-invasive neuromodulation technique that use near-infrared light to stimulate brain activity, and it's worked primarily by enhancing the mitochondrial activity. Clinically, tPBM has been investigated for its potential benefits in cognitive enhancement, neurorehabilitation for stroke and traumatic brain injury, and even as a potential therapy for neurodegenerative diseases like Alzheimer's or Parkinson. So to understand how tPBM influences brain function, we need a reliable method to measure neural activity in real time. So EEG provides an excellent tool for this purpose due to its high temporal resolution. It allows us to track rapid fluctuation in neural activity and analyze functional brain state. So we think the EEG recording microstates are the fundamental unit of brain dynamic. You see in the figure, microstate is a transient quasi-stable scale topography, and it reflects the dynamic activation of large-scale neural network. Microstate can be identified using clustering algorithm applied to EEG time series. So the question is why we have to study a microstate. So microstate can serve as a biomarker of cognitive processes and neurological disorder, and they characterize spontaneous brain activity and transition between cognitive states. Also, a study proved that microstate was associated with resting state fMRI network, and each microstate have a distinct associated with a distinct brain network and have a distinct functional role. For example, microstate A is related to auditory and language processing. Microstate B corresponds to visual processing. C involved in silence detection and executive control, and microstate is associated with attention and cognitive control. And these microstates help us understand how the brain dynamically switch between different cognitive processes. So given that like tPBM has been shown to modulate neural activity, here we have a key question is whether it alter the EEG microstate. So this brings us to our research objective. So we try to solve three key research questions. The first one is, does tPBM modulate EEG microstate dynamic parameter, including duration, occlusion, and transition? And how the tPBM effect, frequency domain characteristic of microstate, and finally, can EEG microstates serve as a biomarkers of neuroplasticity and cognitive enhancement? So we hypothesize that tPBM would significantly alter both temporal and spectral property of EEG microstate, indicating enhanced neural efficiency. To test this hypothesis, we conducted a winning subject randomized sham control crossover study. We have 45 healthy participants from UT Arlington underwent active tPBM and sham tPBM in separate sessions with at least a 1 week washout period. And the inclusion criteria ensure participants have no history of psychiatric or neurological disorder and no condition that may affect cognition. Sorry. For the active tPBM condition, we apply 1064 nanometer near-infrared laser to the right prefrontal cortex at the FP2 location. In the sham tPBM condition, the laser will turn on with intensity reduced to 0.1 watts and a black cap placed to block light. Each session consists of 3 phases, a 2-minute pre-stimulation baseline, 8 minutes of active or sham condition, and 3 minutes of post-stimulation phase. And this randomized crossover design ensure that robust comparison between active and sham condition. We recorded EEG data using a 64-channel biosemi 1010 stem at a sampling rate at 256 Hz. Participants sat in a dimly lit room with their eye closed to minimize external distraction. EEG pre-processing include bandpass filtering from 0.1 to 70 Hz and then a notch filter at 60 Hz. Then we referencing to the average signal and using ICA to remove artifact. This helps to eliminate non-neural artifact such as eyes movement or muscle noise, ensuring a clean signal for microstate analysis. When we obtain the clean EEG data, we apply a microstate analysis pipeline. First, we do in the time domain first, and then we will do in the frequency domain later. So in the time domain analysis, first we have to perform to identify the global field power peak in EEG signal. The peak detected as moments of high neural synchronization. We then use a two-level clustering algorithm to identify the microstate in the EEG data. The first level of clustering will perform to identify the individual microstate for each experiment segment and separately for tPBM and sham session. Then we perform a group level clustering across all subjects to derive 4 dominant microstate. And then the global microstate class will fit back to the EEG data to assign a label of 1 microstate class to every EEG data instant. And and then we calculate the temporal parameter of the corresponding microstate. So the parameter, including the duration, is the average type of microstate remain active. The occurrence is the frequency of the microstate appearance, contribution, the portion of total time spent in a given microstate, and finally the transition probability is the likelihood of switching between microstates in the EEG data signal. Finally, we do some statistical analysis on the result of the microstate temporal parameter among different periods of 2 experiment sessions. This is the overall pipeline for the EEG microstate analysis is the time domain. In the frequency domain, the most difficult stuff is that like the conventional time frequency spatial analysis usually fail to analyze EEG signal with a very short temporal length. In this case, is 60 to 120 milliseconds for the EEG microstate. So because of that, we use another approach using multivariate empirical mode decomposition to decompose EEG signal into a set of intrinsic mode function. And then the intrinsic mode function represent different oscillatory level embedded in the original signal. We then use the Hilbert transforms to was performed on each IMF to estimate the time frequency hybrid spectral of the EEG channel. And then here in this step, we have to use the EEG microstate sequences obtained by the time domain analysis. And then we have to use that on the time frequency spectral we obtained by Hilbert transform to have to generate the microstate specific power chain in the key like different frequency band. So after align the microstate sequence with the spectral, we can calculate the power, the frequency band specific microstate power, and we use do some statistical analysis on the normalized microstate power between tPBM and sham session for different period and different frequency band. For the statistical analysis, in the time domain, we conduct a one-way repeated measurement ANOVA on microstate temporal parameter followed by a post-hoc test to identify significant difference across or experimental period. In the frequency domain, we use cluster-based permutation tests to compare spectral power changes, normalize microstate power as a percentage change from the pre-stimulation baseline. So here's some result of our study. This figure depict the dominant four global microstates across on condition and on participant. And figure B and C illustrate the time-dependent topography under sham condition and under tPBM condition for the across four microstate classes. In the for the time temporal analysis, we observe the change in occurrence of microstate A and D. This figure depicts the occurrence in second of four microstate class throughout different experiment period and for both tPBM and sham session. So here we have the sham condition go first, and then the tPBM session goes second for each microstate, each EEG microstate. So statistical tests show a significant increase in the occurrence of microstate A and microstate D during the tPBM session. No significant difference was shown in the sham session. Another parameter of the temporal analysis of EEG microstate is the contribution. This figure shows the vary of the contribution across four microstate and four experiment period and under two session, sham and tPBM. Statistical analysis show significantly increase in the contribution of microstate during tPBM session. Review that like the degree in the contribution in this type of microstate. We also assess the probability, the transition probability among four microstate class, and we found that there increased transition probability from microstate A to D and from microstate D to A. But there's some significant reduction in the transition from microstate B to C and from microstate C to B. And also no significant change in the sham condition for own own microstate or own experimental period. In the frequency domain, we perform the EEG microstate analysis in the frequency domain and calculate the percentage change in power for each microstate across four frequency band, delta, theta, alpha, and beta. And this figure show the baseline normalized changes in microstate power across the delta band. So in delta band, the delta power significantly decreased across microstate A, B, and D during and after tPBM. This result support that tPBM enhance the wakefulness and cognitive efficiency. And we didn't find any like significant change in the sham condition in dental bank power. The statistical test also shows significantly increase in alpha power in microstave D during tPBM session, as shown in this figure. So no significant difference found in the sham session or in the other microstate glasses. And then this increase in alpha power in microstate D localized from frontal to partial region, and we didn't see any significantly change in other microstate. So based on the result that we obtained from the study, we can say that like there's some like alteration of EEG microstate parameter is due by tPBM. For instance, we have microstate A have significantly increasing occurrence frequency under tPBM. The same as microstate D, we also have the increase in occurrence, and the transition between A and D also increased significantly during the tPBM session. Microstate D have a significantly reduction in contribution during tPBM. And to explain that, we try to see how to explain this finding with the characteristic of each microstate. For example, here we have microstate A is associated with the audio and language processing network, and the increase in occurrence and the transition between A and D enhance in neural activity in the temporal area, possibly linked to a greater neural efficiency. Microstate D is linked to the dorsal attention network, and its increase in occurrence and transition strict tPBM enhances attention control and executive function. Also, the increased transition between A and D may indicate the improved dynamic brain state reconfiguration, which is essential for cognitive flexibility. And finally, for microstate C, the reduction in contribution of microstate D may strike a shift in cognitive processing toward attention-demanding tasks, which align with enhanced neural efficiency in frontal-parietal network. For the frequency domain resolve, we see the significantly decrease in delta power across microstate A, B, and D during and after tPBM. So this can explain that like the expression of delta power on the tPBM strike that we have an increased cortical excitability and promoting an alert and wakeful state. Also is have a potential migration of neural fatigue and allow for greater cognitive efficiency. Delta power expression also support the hypothesis that tPBM enhances neural arousal and attentional readiness, reduce fatigue-related brain activity. And for the increase in alpha band power in microstate D under tPBM session can be explained explain an increase of functional efficiency of the frontal and parietal attention network. Is also explain that tPBM can improve the attentional control and potentially facilitating working memory and executive function. Neurophysiological basic for behavior improvement also observed in tPBM study, for example, increased accuracy of connective tasks, and then this also support our finding as the significantly increase in the alpha band power. The alpha band power enhancement in microstate D also suggest that tPBM optimize attentional processing and connective efficiency by strengthening frontal parietal neural circuits. Also, the result in our study also align with prior study. For example, for the previous study that showed that the tPBM help to increase the functional connectivity, increase alpha band power, and improve the executive function and working memory, and reduce dental power and increase wakefulness. Those findings compatible with the result that we found in this study. In conclusion, tPBM significantly modulate EEG microstate in both time and frequency domain, strengthening and enhance connective function. Future direction, we should investigate a long-term effect of repeated tPBM session, and we can also consider to combine EEG with functional neuroimaging to better map the neural circuits modulated by tPBM. And we can also correlate EEG change with behavior and cognitive performance measure and apply some maybe machine learning model to detect tPBM-induced microstate biomarker. Thank you. So I would like to thank my former supervisor, Dr. Liu, and my colleague at the Biophotonic and Neuroimaging Lab at UJ for their support. And thank you for your attention, and I'm happy to take any question.
Thank you so much, Nghi. If you want, you can stop sharing, although I see quite a few presenters in this list already, certainly very prolific group. So I think we're open for discussion. If anyone has comments or questions or anything to add to what Nghi just presented. I have a question otherwise. So again, the concept of the EEG microstates, it's fairly new to me at least, and wondered how that differs from the model of looking at EEG-based connectivity that also looks at different circuitries, so what is the added value of this measure?
So I think to our best knowledge, I think it's not a lot of study prior study have tried to investigate the effect of tPBM on microstate in EEG microstate, and because we found that like EEG microstate have some like characteristic that can serve as a biomarker of connective process and neurological disorder. We found some other paper that they use EEG microstate to to characterize some brain activity and the transition between connective states. So that's why we thought that why we don't try to investigate if tPBM alter the microstate parameter. So that's why we start to try to link these two concepts and then design the study.
I think we have two questions or comments. I wonder if Nghi, was your comment related to this? Maybe you want to go first, and then Katayo.
Yeah. So let me give you a very quick background in terms of the motivation of doing this. Microstate is also it's been a new concept in my mind back to a few years ago, because it's really getting into depths into EEG signal processing and a really very fast switch in terms of neuronal activity and things like that. So I didn't have the right people with strong mathematical skills who can really have done this work. But Nghi, by the way, she has a PhD in math or simulation from France, and then she got a second PhD with me. So she's very strong in signal processing. So I said, okay, go ahead to try this and then just see what's happening in the brain when we are looking, when we have a light delivered to our brain. And then this majorly 4 states, microstate, which are very fast. It's like a very fast switch from one state to another, either at the resting state or at the cognitive function. So that was the motivation to just learn what's going on and then see, do we see any changes? And then what most of the people who publish papers in microstate was either in time domain, which means you are looking at the 4 states and switching from one to another in terms of frequencies and occurrence and those parameters she mentioned. But then on the other hand, some study did frequency domain. So in her case, she did she combined both time domain and the frequency domain analysis together. So this is kind of interesting results for us to see, or for anyone who in the EEG field to say, indeed, tPBM does change the states. And then now from her result, it looks like A to D, 2 states are switching more, and then that can be related to, as she explained, into 2 different major cognitive functions. So that is the fundamental reasoning for doing this work. And basically in terms of our community, tPBM research community, research-based goals are less, relatively, we have a fewer people doing that versus clinically related research development. So I'm just basically trying to trigger certain discussions or explore more fundamentals for us to understand. So that's the answer to your question.
So just a quick follow-up here. So it seems like the regular connectivity averages the connectivity, the syncing of different brain areas over longer period of time. So while you were able to capture this fast changes in connectivity in showing how the connectivity goes back and forth from one circuitry to the other, and what are the most prevalent within a certain circuitry frequencies of brain oscillations as well.
We are not yet. I'm not completely convinced about connectivity, but more or less just about distribution powers and then from different states. But when you talk about connectivity, this gets to what Cedric discussed last week. When you talk about connectivity, it can be the connectivity along different locations, or connectivity among physiological parameters. And then in her case right now, it can be the connectivity among different states. I mean, microstates, maybe.
Yeah, connectivity between microstates and meaning transitioning from one to the other and so forth.
Yeah, yeah.
Very interesting. I see Katayon, another question or comments?
Okay. Thank you. Do you have my voice?
We do.
Yes. Thank you for your research. It was very informative. And I'm very interested in this field of experience, the increase of or improvement of cognition, because it's related to my field of experience. I'm working in tinnitus, and it's very interesting for me. But I want to ask you, did you think about irradiating to another part of brain and not just to right prefrontal area for improvement of cognition or for modulation of brain neural oscillation? Why did you irradiate only to right prefrontal area and not the other parts of brain?
Nghi, do you want to answer first?
I see a lot of people nodding.
Okay.
I'm so I think like my limited knowledge, but I found that in literature is like prior study, mostly they found the like chain like alteration by induced by tPBM, mainly on frontal parietal area, not one study but like multiple studies. So we may yeah.
Okay, I can help. So sorry, need to cut you short. So basically, when we started, this is about 8 years ago, we took the protocol from Gonzalez Lima, who did a lot of this kind of tPBM work. And then his argument is that right forehead is related to more toward the memory function and reaction time or anything like that. But we follow that protocol, and then we just did all this broadband near-infrared spectroscopy measurement and EEG. But in the recent times, we used this helmet, and then we didn't see immediate effect. So first of all, doing the light from the frontal region is because we don't have a hair to block the light. I mean, that's a very natural argument, but still it's very important to get light delivered because we're getting only 1% of light intensity to the cortex from with respect to the power on the light delivery part. But then with hair, no matter, I mean, at whatever age, some people have a less dense hair, someone has a dense hair, color, and all the things that will affect the effect. So one direct, simple reason is just no hair. It's easy to deliver. And then particularly now from all the measurement that I have, study, we found the light is not only staying in the frontal part when you deliver the light there. It's because of the connectivity. It goes through the whole region of the brain. I mean, from her her data presentation today, it showed that we see the frontal to the back of the brain. So anyhow, that's our initial and current protocol to deliver light at the frontal part of the brain.
Yes, related to laser apple, I know that light disseminate throughout the brain because of the laser apple. But it was interesting for me that you just put it on the right prefrontal area and not the left. And I know that there are some transcranial devices for irradiating all of the brain, and they can penetrate to brain despite the hairs and the dense hairs. They can penetrate because they can increase the power and increase the energy density, and by choosing very useful wavelengths and parameters, they can reach or achieve the light to the right place, because you want to affect limbic system, because it's involved in memory and in cognition, and the right prefrontal area, I think, isn't the place of that we can reach to limbic system.
Now, I can give my intuitive answer. So basically, when you talk about the lymphatic system, that includes CSF. And then CSF can be also part of a shallow layer. I mean, when we talk about the scalp, skull, and then CSF, and then cortex. So in that case, this is something I'll have my current student to do the presentation. We just fixed the time, and that's in March. That's still about 2 months from there. But it looks like light has the effect on CSF. So, I mean, on the dynamics of CSF. But then if you really talk about the center part of the brain to the ventricles or anything like that, that is, I am pretty sure we're not getting there directly from the light in there, unless there's some kind of connectivity effect to get the signal, to get the effect down to the center part of the brain.
Yes.
Yes.
And as Dr. Nicer said, that it can affect the blood, the vessels, the superficial vessels of the brain. So this blood can spread the effect of light. Yes. Thank you very much.
We have a few other questions I see. So one is, how many easily detectable microstates might exist beyond this 4? Any sense of that?
Nghi, you're muted.
Sorry, sorry, I didn't hear the question clearly.
Yes,
Could you please repeat?
do you think there might be other easily detectable microstates that could be measured, for instance, to assess the impact of PBM? And if so, how many?
You mean all the microstate classes, or?
Yeah, A, B, C, D.
Like normally we have 6. Like it depends on the study, but like some study can use 6. But like we limit out in as 4. Like we when we do the clustering, we limit to 4 classes, and then we just choose the 4 classes, like 4 microstate classes, as like, because most like the microstate is the most study, and then they have some study that have the link these microstate with the resting state fMRI network, and they can explain the functional role of these microstates. So that why we use these in our study, but like we can also like extend into 6. There's some study that also use 6 microstates.
So that was a question from Martin. Then we have a question from Huang. So is there any wavelength difference on the effect? I think you only measured 1064, right? So I don't know if you have an answer to that.
We do try different wavelength, right? Like we have some other study, yeah.
With microstates.
Unfortunately, she graduated, and then she could not analyze the microstate for for 800 and 850 nanometer. We had the data, but I have not found people students who can do that. And also I saw another question from
Two
Anne.
more, yes, and next, yes.
Now, because of time, I mean, Nghi, I think I'll just give a quick answer to both of them because the answer is that we don't have answer.
Do you want to read the question or do you want me to read the question?
Please, you can read.
Okay. Were there any non-responders to the tPBM, and if so, why do you think?
So our answer is that we never have done, I mean, never. What I mean is for the study we did, we never did a behavior measure. So we don't know which one you say no responder, which one is responder. So that's why I don't know how to answer that, because that's our future work to combine tPBM with some kind of a behavior measure. Then we can see who is responding better.
Anne is here, but I wonder if she meant people that didn't have the same type of transition in between microstates that typically you observe. Some people might not have it at all. Is that what you
Now,
meant? You give it then.
If that's the case, we didn't look at individual effects, meaning what she did was that we just get this 40 people data put together as a group analysis. That could be something in the future we can look into it to say we got a group level, and then going back to say who are maybe outliers, and then see maybe they're not following the group data analysis. Next one is from...
the last one is, were there any lateralized findings from Fred?
Good question. We don't have answer because we didn't do the analysis in terms of lateralization. So this is why I think it's good to have other groups who are doing some work, then we can compare notes. But so far, we're just basically give some preliminary data or preliminary results, and then just trigger the community and see whoever has interest, then we can later compare notes. Thank you very much.
We have a few more minutes if there are any other questions or comments. All right, so I will leave you all to your own microstates, and thank you all. Thank you so much, Nghi, for this very intriguing models, and to be continued. Take care.
Thank you. Nghi, thank you very much.
Thank you, Dr. Liu. Thank you so much.
Thank you, Dr. Truong.
Bye.
Bye.